Hi, everyone. Welcome back to another session of the Cambridge MedAI Seminar Series. Our first speaker today is Dr. Delshad Vaghari. He's a postdoc research associate working at the Adaptive Brain Lab at the University of Cambridge. And his research uses machine and deep learning to study neurodegenerative diseases. His main interests are the interface of AI and brain sciences. And he's also interested in the use of a broad range of neuroimaging techniques such as MAG, MRI, PET, etc., to study dementia. Currently, he's working on developing AI models for the prognosis and diagnosis of dementia and drug development. Welcome, Delshad.
Thanks, Ines. Okay. Yeah. So thank you so much for inviting me for this amazing seminar series. I'm very pleased to be here today. Today I'm gonna talk about robust and interpretable AI guide marker for early dementia prediction, specifically in real-world clinical setting. Sorry. Yes. So just a bit background. Excuse me. Yes, it's now shown. Yeah. So here is the hypothetical progression of dementia on x-axis, and we have normal to abnormal on y-axis. If you look at the brown line here, the clinical function, we can see people who are suffering from dementia. Clinicians give them dementia label because for sure they surpass some criterion. But if you look at the purple line, there are people who, if we do more subtle tests, like cognitive tests, they cannot pass them, but they can cope well in everyday life. We call these people mild cognitive impairment, or MCI, which is believed to be a predominant stage for dementia. Other colors, like red one, there are brain measures that we usually capture from brain imaging. For example, the red one here is PET, that can measure amyloid. Using imaging techniques like PET, we can predict dementia decades before symptoms, between showing symptoms. So predicting dementia at early stages is very important, it's vital. Yes.
Yeah, so this it's not
Just to show with mouse. Yeah, okay. Yeah. Yes. Yeah. Different studies shown that if we are in the earlier stages of dementia, the treatment might be effective. So here on the left, we can see a brain MRI for a healthy individual, and on the right, a patient with Alzheimer's disease. But just by naked eye, we can distinguish these two MRI. As you could see here, in Alzheimer's disease, we have a huge reduction in gray matter density. But the stage that we are interested in is MCI, or mild cognitive impairment. We cannot see just by looking at the MRI whether someone has MCI. So we bring an AI model to answer this question for us. So when we use machine learning, we face with three main challenges, especially in clinical settings. First one is, can we make AI to be biologically informed and clinically relevant? The second challenge is, can the model be interpretable? Or in other words, can we understand disease mechanism? And the third one is, can AI be generalized to global solutions? In this study, I'm going to address these three challenges with different solutions. So we usually need more than one single data or modality to train our models. So we can train the models with wealth of data. Here in this specific study, we focused on cognition and brain scans. We used some cognitive scores and some features from brain MRI. But of course, we can use other markers like blood markers or genetics for training our models. So today, I'm going to talk about how we can use these biologically or clinically relevant features to train a machine learning model, which is based on metric learning, and how we can extend this model for prognosis and trajectory modeling. So the framework we use here is generalized metric learning vector quantization, or GMLVQ. GMLVQ is a part of a family of learning vector quantization classifiers. This type of classifiers operate in a supervised manner. It iteratively modify the class-specific prototypes and learn boundaries between classes. For example, here we have class 1 and class 2, and for each class we have one prototype, P1 and P2. During the training, when we fed the classifier with training examples, the prototype closest to each class is specified. Prototypes are then updated so that the prototype defining the same class is moved toward the training example, and the prototype representing the different class is moved further away. So generalized metric learning vector quantization is an extension of LVQs. It uses actually a full metric tensor for more robust distance measure. Applying a metric tensor allows specific feature scaling to account for different feature scales. So after training, we will end up with having two prototypes, P1 and P2 for each class. So for any new example like X, the classifier calculated the distance to each prototypes, and if it's closer to prototype 1, we assign class 1 to this example. So in the first step, we train a GMLVQ for classification of stable MCI versus progressive MCI. What is stable MCI? So we follow MCI patients for 3 years, and if at the baseline they are MCI, if they remain MCI within a 3 years window, we label them as stable, and if they converted to Alzheimer's disease, we label them as progressive MCI. So the first step in this study is to train a GMLVQ to classify these two groups. So next step is to extend the classification problem for prognosis and trajectories. Here we use a technique called scalar projection. If we have, for example, the prototype for class 1, the red square here, and the prototype for class 2, if we consider a vector here, what actually scalar projections do is measuring the distance to one of the prototypes along this vector. So by using this technique, we can have a prognostic index depending how far we are from the stable prototype, or the prototype belongs to stable MCI class. We can decide whether they remain stable or they will be converted to Alzheimer's. We even improved the model based on ensemble learning concept. We trained 400 different models using random sampling with resampling. Then we select top 20% of them based on in terms of accuracy of these models. And we predict whether someone is PMCI or SMCI based on top 20% of 20% models. So each of these models has two prototypes, one for stable class and one for progressive progressive class. So when we have 80 models, which is the top 20% of these 400 models, we take average of prototypes among these top 20% of classifiers. So for training data, we use ADNI dataset, which is a research cohort. We use, as I just mentioned, we use longitudinal labels to train our model. And then we test our model in 3 different samples, test sets, actually. One of them is another part of ADNI, which is a research data, and 2 cohorts, one from UK and another one from Singapore, to see how well our model can be generalized to real-world data. So one of the first challenges is interpretability. Using GMLVQ, we can have a metric tensor. Here we train our model on some cognitive scores like ACE, which is Addenbrooke cognitive examination, MMSE, mini mental state examination, and a feature from MRI scans, gray matter density. After training the model, if we go to core of the model, we can see the metric tensor. The metric tensor, the diagonal elements tell us which feature is the most important one. For example, here we can see ACE memory is the most important feature for classifying PMCI versus SMCI, and then it is MMSE and gray matter density. The performance of our classification model was 81%, which is actually typical in literature of dementia. Of course, we can train our model with more expensive imaging features like PET data or genetic data like ApoE, which is a genetic risk for Alzheimer. If we use these kind of features, the accuracy of model will be 90%. And we can see from diagonal elements which feature is the important one. And off-diagonal element of these matrix tell us how this feature interacts together. So we extract this prognostic index from a test data, which is a research one at the beginning. So this prognostic index, which is actually distance to stable prototype, we can divide them to 3 different groups: stable, slowly progressive, and rapidly progressive. It depends how far they are from stable prototype. We have the true labels for these patients or individuals. So we can see that most of patients with Alzheimer, our models stratify them as slowly progressive or rapidly progressive. And people in early stage of Alzheimer, our models stratify them as slowly progressive. And healthy controls, the models stratify them as stable. We also train some pre-symptomatic individuals, people that do not have any symptoms. And our model also can stratify them to these 3 groups. So the next challenge was, can AI be clinically relevant? Here we tested this prognostic index with different measures. We see here that the model-derived prognostic index is highly correlated with cognitive decline, with increasing amyloid, with decreasing gray matter, and also it can split patients based on ApoE4. The last challenge was actually can this model be generalized to real world data? So first we tried a cohort data from memory clinic in UK, Cumin MC. Our models stratified them to stable, slow, and rapid. As you could consider here, we have also the real labels to test whether our model stratified them correctly. So we can see that those that our model, most of them, our model stratified them as rapid, they are Alzheimer's disease. And MCI patient, they are slowly progressive. And other dementia subtypes, they stratified by our model as rapidly progressive. And people with memory symptoms or depression, for example, they are stable. We also tested this model on a data set from Singapore. This data, MAC data set from National University of Singapore. The model again stratified them to these three groups. And here again, the rapidly progressive patients are Alzheimer. The moderate MCI, which is a progressive stage of MCI, they are most of them stratified as rapid, and the early stage of MCI or mild stage of MCI, our model stratified them as slow. And healthy controls, they are stable. Our model stratified them as stable. We also performed longitudinal validation. We performed survival curve. For the MAC data or data from Singapore, we have longitudinal information. So we can see whether our model, when our model stratified them to stable, slow, and rapid, how they will be over time. Here, over a period of 5 years, we can see when the model at the baseline says someone is stable, they remain stable over 5 years. And when the model stratified them as slowly progressive, the chance of converting to Alzheimer after 5 years is 50%. And when the model stratifies someone as a rapidly progressive over a period of 5 years, the chance of converting to Alzheimer is around 80%. And finally, we look forward to change the clinical pathway by assigning the patient the current labels, stable, slow, and rapid. We can tailor the care pathway. So imagine a patient go to a clinic by taking some cognitive test and also having an MRI scan. The model can stratify them to one of these groups. And based on this information, the clinicians, it may help clinicians to decide whether they need a PET scan or whether they need to be hospitalized. And at the end, I would like to thank my colleagues and collaborators. It was an amazing collaboration. And thank you for your time.
This also gave us the basic element of the data we use in computational pathology, which is host-led image. These are the products of creating a single high-resolution digital image from a glass slide. They are usually really high resolution, really big. One image can reach several gigabytes, and they are usually stored as image pyramids. At different resolution level, you will actually be able to find different features like cellular features or tissue level features or the spatial organization of different kind of cells. So very rich information is incorporated in this kind of images. And naturally, like it's very time consuming for pathologists to go through the whole massive image to find the small regions of interest on their own manually. So this is exactly the task where AI can help a lot. So this is the standard workflow we have in building AI models for computational pathology tasks. Essentially, I believe like the goal of this computer vision model for computational pathology is to learn a vector representation, you can call it embedding of the whole slide image, and then use that embedding to predict all sorts of downstream tasks, may it be survival analysis, may it be disease classification, etc. So the standard workflow after you get a digital image, the first step is usually like identifying the location of the tissue and chop them up into tiny pieces, which can be digested by normal computer vision models. And then given the high variability of histopathology images, usually like having some quality control to remove the artifacts and some image normalization or augmentation can potentially improve the model performance quite significantly. An important element of computational pathology models is the interpretability, because eventually, well, black box models work for certain tasks, but when you're applying AI models for biomedical research, you actually want to know what factors are driving the model to make certain predictions. And then you can validate your finding with pathologists to see if a model is actually learning something biologically meaningful. So mechanisms such as attention or saliency maps can at least help us have a broader view about where exactly are the morphology that the model is paying attention to. So having said that, like, I guess like I just want to complain about certain challenges I have encountered with developing AI models for histopathology images, especially histopathology images from real world settings. So the acquisition of the digital image was the first step in the previous workflow I have shown, but actually there are so many complicated steps involved before you can get your digital image. You first have the like anatomical acquisition of the human tissue sample, and then you have to go through this histopathology lab routine to get the glass slide. And then you need to use different types of scanner to scan the glass slide to finally get the image. During each of these steps, all sorts of artifacts can be introduced. I've seen a lot of weird stuff I never expect to see in biomedical samples, but there they are. Well, some of the artifacts are actually trivial. Some of them can severely impact the usability of your image. So if you don't remove these images from your data set, you're basically feeding a model garbage. And again, like a more tricky issue with histopathology images from real-world settings is they are usually not properly sorted. They can come quite messy. You can have different types of tissues mixed together. Even if you have tissues from the same region, the same organ, they can be acquired using different techniques. Sometimes you have surgical resections where you have a nice comprehensive overview of how the inside of this region looks like. But most likely in clinical settings, the first step is something less invasive. So you use core needle biopsies a lot these days, and then you end up having a very tiny, very partial local view of the region that you want to analyze. And then again, like even though, well, I think when we talk about computational pathology, most of the models are developed for HND stent images, which is the most standard one, like hematoxin and alcin. But when you are looking into specific organs, there are actually special stents used to highlight different structures on tissues. So something trained on HND can hardly be applicable for something. For example, here, what we are showing is an example of a kidney image stent with Jones silver, where the basement membrane on the glomeruli are highlighted with this black color. And that is the kind of color you would not be expecting to see on an HND stent image. And even if you have everything controlled, even you have the same tissue, the same disease you're studying, the same assay you're using, simply just using a different type of scanner, like asked before, like a different brands of scanner give quite different appearance, even if you're scanning the same glass slides. So here you can see an example, just using these 4 quite commonly used different types of scanners can lead to quite different color profiles. And this can cause quite severe domain shifts in computer vision. And another thing is, like, when you develop an AI model, you want, especially if you want to ever apply your model in clinical settings, you need to make sure it's very prone to noise, because it's commonly known that computer vision models for diagnostic purposes sometimes can be very prone to adversarial attacks. Some imperceptible noises can lead your model to make unrational wrong decisions. And that's exactly the case where we want to avoid. And again, like I have mentioned this before, but I want to tap on this again. Interpretability is very important in computational pathology work because you want to be able to validate with the pathologist where you find interesting. And also, if you are interested in looking for novel digital biomarkers from the tissue, you actually need to be able to know where your model is paying attention to, what is driving your model to give certain decisions. And here, like simple techniques like attention mechanism can give us a lot of insights into what sort of cellular structure or what sort of cell population can be correlated with a specific disease outcome. And yeah, like sometimes your model will even be able to pick up something that cannot be so easily noticed by pathologists with their naked eyes. So it's actually quite useful. And finally, some examples of how we are using real-world histopathology data sets to support our research. So this is a brief overview of the 3 real-world data sets I have used while working at AstraZeneca. This is just a like partial snapshot of what we have worked with. Not like I'm sure there are other cohorts that are using as well. So first we have Tempus. Tempus is a leader in translational oncology research. They basically provide multimodal data to including multi-omics, including histopathology images and clinical records to support oncology research. But I want to highlight that it's commonly, this kind of precision medicine research regarding omics or images are quite commonly used in cancer research, but less prevalent in study regarding other therapeutic areas. We are also using similar approaches by developing AI models to look into histopathology images collected for inflammatory bowel disease, or in this case, chronic kidney disease. I would like to share more about my work on Nurture. This is the first kidney biobank that covers multiple centers across England, Scotland, and Wales. It's also one of the first multimodal kidney biobank that has ever been generated. So we are actually using different approaches to analyze the renal biopsies we got from Nurture, the CKD, aka chronic kidney disease cohort. So first, and maybe even the most crucial step is to have a quality control tool to remove the low quality image you get quite often from clinical settings. So we have a QC tool that can help us like filter out the unsuitable images, and then only the images that can pass this quality assessment will be passed to downstream analysis. And my specific work involved in the segmentation of relevant tissue components, in that case, the glomeruli on the kidney cortex. In the meantime, we also have a generative AI model based on cytoGAN. So the idea is to generate kidney-specific spatial stans, such as PAS stan or Mason's trichrome stan from the standard H&E. And again, like we are also using weakly supervised classification and multiple instance learning to directly infer disease progression or clinically relevant outcomes, such as the necessity of kidney transplant or dialysis, based on the snapshot we get from the renal biopsy. So it's important in the diagnosis for chronic kidney disease to have a renal biopsy, because usually the visually perceptible changes are happening on the glomeruli and the tubuli, and these are located in the kidney cortex. The standard way to get this kidney biopsy is to use a core needle to get a tiny piece of tissue from the kidney cortex and then stain it with, well, like commonly used acids would be periodic acid Schiff or Mason's trichrome or Jones silver. These are the most commonly used ones for kidney tissues. And here we focused on past stained kidney biopsies simply because we have mostly this assay in the Nurture cohort. Well, the first task I got was to segment the glomeruli because it's a very important functional tissue unit on kidney. In simple word, basically you have blood with waste flowing into the glomerulus, and then they will get filtered. The waste will be kept there, and then you will have clean blood without waste going back to your blood circulation. And the pathologist actually will look a lot into the glomeruli to analyze basically the stage of chronic kidney disease and to evaluate how much kidney function the patient has already lost. And eventually they can even predict whether there will be potential of death or like an inevitable need for kidney transplant. So this is a very informative part on the whole tissue where they are looking into. So we thought it'd be nice if you can have a tool to directly locate the glomeruli and extract some features from these glomerular regions automatically to support their downstream analysis. Initially, well, we thought it's going to be, I thought it's going to be a simple task because it's not a new task. It has been published before by many papers as well, and we did find a publicly available model. The problem is the model was trained on resections from reference kidneys. So these are basically healthy glomeruli. They look nice, they are neat, and they're easy to detect. But when you are, in our case, we got this tiny needle biopsy of sometimes incomplete glomeruli, and a lot of them are severely diseased. Well, like they don't really look like what they are supposed to look like if they were still functional. So in that case, naturally, the model trained to identify the healthy ones failed to detect or segment this diseased glomeruli. But this is exactly what we want to find on these kidney biopsies. So our idea is to somehow find a way to make the segmentation model learn from our unlabeled clinical data to pick up this disease morphology on glomeruli. The problem is, well, if you have worked in medical imaging, I believe you must share my opinion. It's really difficult to get annotation from experts. It's just very time consuming. It's also really hard to convince someone to manually annotate your stuff for several hours without any concrete outcome on their own. So like we wanted to find a workaround to bypass the need to get pathological annotation first. So that's why we applied the semi-supervised learning approach. Since we have some publicly available annotated data set, we have our unannotated biopsies. So we were thinking, why not just use both of them? And this is a simple idea, actually. To train this semi-supervised learning model, we have two branches. The first part is the standard supervised branch. So that's basically how you train any computer vision model. You have an input image, you pass it to your model, you get a prediction, and you compare your prediction with your ground truth, and you calculate your supervised loss, like standard stuff. And then for the unlabeled image, it's more interesting. The unsupervised branch works that way. First, you have your, sorry, you can see the later one. First, you have your input image, which is not labeled. And then we will run some weak augmentation, such as flip or rotate, and then pass it into the computer vision model and get a prediction. And this prediction will be used as a pseudo ground truth, like a pseudo label. And then based on this, we will run some more augmentation of the data. So the first thing we did was to take the embedding you get from the encoder, run some feature perturbation, and then use the perturbed features to predict, to get another prediction on this perturbed data. And then similarly, we have two strong augmentations. For each strong augmentation, you pass it into the model, and then the model will give, respectively, will give predictions on these different augmentations. And then you compare your pseudo ground truth with these predictions you get after different augmentations. And then you calculate your loss this way. The idea is if the model is actually picking up meaningful morphology on the image, the augmentation feature perturbation shouldn't change the prediction that drastically. So you should be able to make similar, at least similar segmentations on these different inputs you gave. This is a way to utilize unlabeled images without having to get annotation first. I should mention that we didn't really develop this thing on our own. It was some like pipeline developed on natural images by other people. We just simply adapted in a way so that it works on histopathology images. And another bit of this work is that we replaced the standard state-of-art segmentation model UNet in medical imaging with SegFormer, which is a lightweight vision transformer based model that has only 10% of the trainable parameters compared to the standard UNet. So we have seen some good results on the 3 external data sets. These are all external validation data sets. We haven't shown the model during the training process. So we can see on one of the data sets, we got actually a quite drastic increase on DICE score, and yeah, like a pretty decent increase on recall. For our clinical data sets, this kidney precision medicine project and nurture, unfortunately, we didn't see a significant increase on DICE, but it's actually quite promising to see an increase on the recall because the previous model struggled mostly with identifying the diseased glomeruli. And we believe this recall rate was increasing because the model is getting better, at least learning to gradually pick up these disease morphologies. Here you can have a better view of how it works. Well, actually it's a bit confusing. So the first one is the ground truth, and the green one is the, the first one is the input image. Second one, the green one, is the ground truth label made by pathologist. Third one, the yellow one, is the prediction made by supervised model. And then the blue one is the prediction made by semi-supervised model. So when you have a fairly healthy glomerulus, it's actually not that difficult a task. Both models performed well, but we can see actually when the glomerulus is showing certain disease morphology, the semi-supervised model is getting much better at picking up this instance. For example, when you have this mesangial cell overproliferation on certain glomeruli, the supervised model failed to identify the cases, whereas the semi-supervised model picked it up. Though not perfectly, but at least like it managed to detect here is something that we are interested in. And also like when they are showing local changes, for example, on this case, when there's a local sclerosis on this glomerulus, the supervised model decide that the rest of the glomeruli, the fairly functional part is a glomeruli, but the semi-supervised model managed to include the sclerotic part as the glomeruli as well. This is important in a way that because we also want to extrapolate features such as the volume, the size of glomeruli from this detection segmentation results. So it's actually important to pick up the whole thing instead of just a snapshot. And I put these two here because I want to support my argument that real-world histopathology images are of pretty terrible quality. Because here we have example where the ground truth label shows that this is a glomerulus, but it's clearly a mistake. And here, I believe if you are very perfectionist, you probably will discard this image because you can see this massive air bubble on top of it. But well, I'm still glad to see that the semi-supervised model managed to detect this glomerulus despite this massive air bubble on top of it. So it's not perfect yet, it's still work in progress, but yeah, at least we are seeing that the model is learning more disease morphology. So the next step of this work might be correlate these glomerular features to more clinically relevant outcomes for chronic kidney disease, such as the estimated glomerular filter rate, or to estimate the speed of progression for these patients. The last bit was, I would like to tap on certain future perspectives I have for AI in computational pathology. So having worked a lot with real-world histopathology data sets, I feel it'd be crucial for the community to have a shared open source quality control tool, which can be applied to histopathology images. And also, since sometimes we have consecutive tissues stand using different assays, it'd be nice if we have an image registration tool that will allow us to correlate cells and tissue components from different assays with each other. So it will give you a more comprehensive representation. For example, you can correlate your IHC with your multiplex immunofluorescence image, or if you correlate your HND with some more fancy types of image, that would be quite interesting. And again, this is a buzzword, but yeah, a lot of people are talking about foundation models. So I think, yeah, it's promising to see if we will get a real foundation model for histopathology. Actually, you can look into the publication made by Faisal Mahmoud's lab last week. They actually published 2 foundation models for computational pathology. It's worth a read, I would say. And again, I believe histopathology image is a cheaper and more promising way to incorporate the spatial nature of tissue and disease into your biomedical research. Compared to more spatial techniques like spatial transcriptomics, this is much cheaper, and hence, like, I think there's much more potential in developing something for clinical settings. Yeah, that's it. Thank you. Thank you for having me.